So my name is Tyler Patty, and I did my research project on Asian carp. I've actually been researching these fish for the past 2 years, and essentially what I've been doing is I've been investigating the hair cell density differences between the silver and the big head. Main reason behind this is because the both these species are invasive species, and they have been affecting the Mississippi River drainage system. So there's actually been quite a bit of promising results that show that you can use sound deterrents to push these fish out of the way. Um, Part of this research has kind of shown that one fish will respond better than the others to this sound. So that's mainly what we looked at. So the title of my project is Differences in the Inner Ear Hair Cell Densities May Explain Behavioral Responses to Sound in Two Invasive Carp Species. So basically, we start off with what are Asian carp and how do they hear? So the Asian carp, the two that I studied are the silver, which is pictured here, and the big head, which is pictured here. So the silver, its name is Hypophthalma ichthyes molitrix, and the big head is Hypophthalma ichthyes nobilis. For just making things easier, I'm going to refer to it as silver and big-headed carp. So essentially, the fish, they have two ears, just like us. But their ear is organized just like this, and this is actually an ear from a big head carp. And the carp they belong to an order of fishes called the autophysins. And essentially, what autophysins are is they have these special adaptations called Weberian ossicles, which is basically bones that connect the swim bladder to the inner ear mechanically. So basically, what happens is the swim bladder will receive sound from the exterior, and then it translates that sound to these Weberian ossicles, which then transmit that sound to the inner ear. So this basically what's happening is you're having a conversion of auditory information to mechanical information, then to neural information. So essentially the way it works is the fish have these stones, and they're called otoliths, that sit inside their ear. So they sit inside each ear. So you have the three ears of the saccule, which this is the saccular otolith, the utricle, this is the utricular otolith, and then the lagina, which this is the laginar otolith. So essentially, these otoliths sit atop hair cells, and then when the sound from the uh, is transmitted from the burial ossicles to these inner ears, it causes those stones to move back and forth. This movement causes these kinocilium, which are pictured right down here, to move back and forth. And the hair cell bundle consists of the kinocilium and a bunch of stereocilia. So the kinocilium is really the one that's of the most importance, because when it moves back and forth, that determines whether it's hyperpolarized or depolarized. So essentially, this whole advantage of the autophysins is you have sound that starts off as auditory from the swim bladder, is then transmitted as mechanical through the Weberian ossicles, which is then transmitted to the ears, which is then transmitted to the brain and a neural. So you have all this conversion of the signal. So basically, this lets the fish hear, which is very, very important. So why is this hearing important? Well, this connection is why they have this behavior, or this is why we predict they have this behavior. So like I said before, the fish, they jump out of water very sporadically. This sporadic movement is only by the silver. It's not by the big heads, which is kind of what piqued your interest to research the ears anyway. So just how you can determine how the range of hearing that a fish has is you can do what's called an auditory evoked potential. I did not do this, but this has been done. But this is how you can determine how you can how you determine the range of the fish's hearing ability. So essentially, they just stick electrodes inside the fish's brain while it's still alive, and then measure the frequencies of sound, or measure the actual potentials that it responds to given certain frequencies of sound, to try and determine a range. This is important because this you want to know what range of sounds these fish will respond to. So what did I do specifically was I looked at the hair cell bundles inside the fish. So these hair cell bundles are Like I said, they're on the saccule. So what I did was I stained the tissue with a secondary antibody, fluorescent antibody, and then took it and imaged it under an epifluorescence microscope, which is where you get this image right here. This is actually a big head saccule itself. So basically, all these little white dots are hair cell bundles. So you have to take an image at an extremely large magnification, and then I took all those images and then I pieced them together. That way you can get this whole image at an extremely large magnification. So after I did that, I took 9 2,500 micrometer squared boxes and I placed them throughout the saccule at strategic locations. Those locations are diagrammed here, and they are from rostral to caudal ends. So rostral meaning the head end, caudal meaning the tail. So basically these boxes just stand for percentages, 5, 15, 25%. And basically that's just the percentage of the saccule at that point. So what I found was that the silver actually possessed a higher hair cell densities at the 55 and 65% locations, pictured right here, than the big-headed did. So this might be the reason for why they jump out of the water and the big-headed don't. So again, that could be a potential reason. So some more evidence for that is Dr. Smith, actually, my research mentor, he actually did a paper on the tonal topic organization of goldfish themselves. Why this is important? Goldfish belong to the same family as carp, the cyprinid family. And they actually found a tonal topic organization for a goldfish. So tonal topic organization, all it is you have certain frequencies of sound that coordinate to certain areas of the cell of the of the saccule that detect those sounds. So it would make sense that maybe the silver have a greater tonal topic organization at the central locations compared to the big head, which might explain why they jump out of the water. Because as I said before, they do have a higher hair cell density at these regions. And if they are tonal topically organized just like goldfish, which is highly likely, then that might explain why they act differently than the big-headed does. So some questions you might have about about my research. What about the other 2 ears? Because as I mentioned before, the fish has 3 ears. And you're exactly right. The saccule has been researched a lot, and it has been found to be primarily an auditory organ, and the utricle has been found to be primarily a vestibular organ. And the lagina kind of serves as a intermediate. It kind of helps both organs. It kind of coordinates both organs. So that's why we focused on the saccule, was because that has been proven time and time again to be an auditory organ, and the other two haven't. I have personally worked with 3 types of fish in Dr. Smith's lab, and I can personally say the saccules look very different among species. So I can say firsthand that they are extremely different. So that's why we did it. Another question you might have is, will these certain sound deterrents that we come up with, will they hurt native species? They possibly could, which is why we're doing extensive research to make sure that that doesn't happen, because we want to get rid of the carp, but we don't want to hurt the native species. So we're doing all this research to kind of gain an understanding of how they hear so that we do not affect our native species and we can get rid of the non-native. Another question you might have is, okay, so we determined that this hair cell density difference might be the reason why the silver jump and the big head don't. What about the big head? What are we going to do about it? That's a great question. That's future research. Okay, we have found a difference between the saccular hair cell densities at the 55 and 65% location between silver and big head. So now we need to investigate, okay, maybe let's look at the other 2 organs. Maybe the big head have a higher hair cell density at the other 2 organs, or in the other 2 organs, and that might be a point to look at to see, okay, is there a difference here? Can this be used to develop deterrence for them? So that's an excellent question, and that's for further research. So that's definitely something that we need to look at. These are my references from my images. I appreciate you listening to my presentation, and I hope you have a good rest of the day.